let's get going. We're going to have a lipid kind of morning today, and I'm going to give you a very brief primer on lipid biology. It's not meant to be comprehensive in 15 minutes, but just to get the language down. Basic molecules in human plasma, lipid by far is the most abundant class of lipids, and this is probably an underestimate if you actually look at how many different species that we can find in plasma compared to how many species of different things that we can find. I think it's lipids are really prevalent, and they turn out to be fairly easy to measure in a shotgun manner. TM will go through that, and they seem to be really great biomarkers of a lot of processes. And I think you saw a little bit of that already. So here's a definition of a lipid, amphiphilic, small molecules, carbon, you can talk about your chemistry. This is the chemical definition. There's a great resource from the UC Davis Metabolomics or Genome Center, but it's actually done by Ollie Fine's lab, and they have a great sort of lipid blast project, and there's a lot of resources within that and linked to that you can use to look up your favorite lipid and lipid pathways. There's also the help me, TM, the UCSD. I think I have it in here. They have the lipidome project as well. All right, so there's classifications, and a lot of them classified on biosynthetic routes. The categories, the big categories of the ontology of lipids are fatty acyls, glycerol lipids, glycerophospholipids, sphingolipids, saccharolipids, and polyketides. We measure the top four. These we really haven't gotten into yet, but there's a lot of them in there, and it's probably the next area of biomarker discovery, the sugar lipid conjugates, because they really form a lot of the complex cell surface things, and they probably will give you some idea what's going on in cells. And then there's the steroid lipids and the prenol lipids. These are a subclass of lipids, the carbocation-based condensation reactions that form these kinds of lipids. And the big one is the sterol lipids that most of you know about. So the lipids that are in there, it turns out that the sterol lipids, cholesterol esters and things like that are the most common that you find. The glycerophospholipids and the glycerol lipids are the second most common that you find. Fatty acids are actually a very small fraction, and sphingolipids are surprisingly a larger fraction than just even the free fatty acids that you find in the blood. And so if you do GC mass spec, and this is just one list, I think we hit a lot of these kinds of things, but you can get, and this is out of this lipid maps project, that you can actually find the number of compounds that are sitting in each one of these. And so we're talking upwards of 200,000 different lipid species that can be identified in plasma. So it's complex. There's lots of them, but they can really serve as really interesting biomarkers. And I think I talked about that a little bit yesterday. It's probably going to be the best thing for dietary assessment of macronutrient, because these things because they cross all these different metabolic pathways. All right, so a fatty acid, it's pretty simple. You all know fatty acids, palmitic acid, C16 lipid, has a carboxyl group on the end. It can be esterified. Usually this floats around in the blood as a free fatty acid or esterified to glycerol or other hydroxy groups in different kinds of compounds. So the ester group is what's there. There can also be ether groups here, so this double bonded oxygen can be missing, and there's probably lots of these ether-linked lipids in the blood. I think it's those are quite common in the blood as well. So your common fatty acids, you've all heard of palmitic acid, stearic acid. So these are the saturated fatty acids. This is of the fatty acids that circulates C16 with no double bonds and C18 with one double bond. The oleate are the most common fatty acids. And then there's two, linoleic and linolenic acid. Those are the ones that we can't make that are essential fatty acids. And you need, when you're growing, you need about 2% of your caloric intake needs to be fatty acids. Most of you in the audience can go for probably a couple of years without having to eat another fatty acid before you start suffering from inefficiency. The thing is that we just can't put it in this double bond here, and so we need to take that in to be able to generate a lot of the cosanoids and other signaling molecules that are generated from these essential fatty acids. Triglyceride is 3. These are lipids. The glycerol backbone. Mahmoud actually showed you some of the synthetic pathways and how you can actually look at the synthesis of the. You can follow the synthesis using either a glycerol tracer or glucose-derived glycerol tracers inside the cell, or using labeled fatty acids to follow the synthetic pathway. For all of these fatty acids that are esterified to glycerol backbone, we can see a lot of those, and we use those a lot to look at intracellular metabolism of fatty acids. As you know, when you get a fatty acid, and we use this, the elongation desaturation that occurs inside, mostly in the liver, but some of it happens in the adipocytes, to follow metabolism, and it can actually, again, be a biomarker of sort of nutritional status, and these are affected by some classes of drugs, some of the lipid-lowering agents, but mostly it's affected by diet. So again, the desaturase, which humans have one of in the liver, SCD1, is induced by carbohydrate feeding. And so this is a really good marker. The ratio of the C16-1 or C18-1 to C16-0 is a good marker of how much looks like how much carbohydrates. And the elongation enzyme is also a regulated enzyme. And some of these elongases have genetic allelic variations that are important, or looks like it's important for different disease states. So the FADSs may be good, maybe an important allelic variation that control the generation of a lot of intracellular signaling molecules. I doubled that slide. Sorry. So the glycerol lipids are derived from these keto-acyl groups, and so this linkage here, and the isoprene units, these are intermediates in cholesterol synthesis, but they also are important molecules for linking proteins to different subcellular locales. So the glycerol phospholipids are, glycerol with a phosphate head group. Then the X is the wild card there, and TM will go through, and we can measure. We can see a lot of these different head groups and use it to quantify the different kinds of lipids that are there. The important thing is that there's some rules that if you're what's called the SN1 position, when you have a phospholipid and you're synthesizing these phospholipids, usually the SN1 position, which is the first carbon in the glycerol side chain, so this is historically the third carbon here is the SN3 position. This is usually a saturated fatty acid, and usually this is an unsaturated or polyunsaturated fatty acid in the SN2 position. So when you have these chains and you actually look at them, you can make some assumptions about what might be in the first position, the second position. And it's interesting if you actually start thinking about using these kinds of information for tracing studies, and you want to look at either the generation or the destruction of different glycerol phospholipids by looking at what you get basically the phosphatidic acid, which has the one carbon on there. You can actually make some inferences about where things are coming from and flux through different pathways. And so it just takes some skill. But if you flood in a lot of palmitic acid, or saturated fatty acids, and you look and use that for flux, you clearly will see SN2 incorporation of the fatty acids. Of the saturated fatty acids in SN2, and if you really deplete the cells of saturated fatty acids, which you can do, you'll get unsaturated fatty acids put in the SN1 position. The sphingolipids are very interesting compounds. They have clearly a lot of signaling capacities. Ceramides, as a lot of you know, are probably important for some apoptotic or toxic pathways. The cerebrosides there's a lot of inborn errors in metabolism and a lot of these different things that causes a lot of different disease states, and they're becoming very important. Markers, again, of different inflammatory states or different metabolic states. The cerebrosides, that's what Shaman, that pathway. So actually it's helped the University of Michigan. A drug was just approved that was developed by Jim Shaman and his team here at the University of Michigan for treatment of Gaucher's disease. And so so that's actually bringing some money in that's really been helpful to a lot of people in the medical school these days. Cholesterol esters are, again, one of the more abundant lipids in the blood, but also in the cells. The biological properties of these, except for being biomarkers, is unclear. I'm not sure there's a lot of signaling. There may be some role for cholesterol and cholesterol esters in signaling how much cholesterol is in cells and releasing sort of enzymes to activate cholesterol gathering pathways. But as far as signaling, I'm not sure that there's lots known about what they do besides sort of carry cholesterol around. Bile acids are derived from the cholesterol that's in the gut, and again, there's probably 30 plus of these. We can measure a lot of these both in the plasma and in the fecal material, made in the liver. Obviously, most of you know about that. And then steroid hormones, again, are also based on cholesterol and the important pathways in the adrenal gland and in the reproductive glands. To generate really important molecules, and disrupting these pathways are obviously have physiological consequences. So this one's really easy to read. I have the reference there, but this actually, when you look in blood, where are these lipids? What particles carry them around? And so when you do some of these measurements, usually when you just extract a lot of lipids, what you see is a lot of different kinds of lipids. But lipids in the blood have to be carried around by lipoprotein particles, because otherwise you get soap floating around your blood, which is not good for you. And so this gives you an idea of what the relative concentrations, but it turns out that the relative concentrations of different lipids within these particles are going to be important for different pathological states as well. And so, subfractionating these particles and seeing what's where, when is going to be one of these games that we play when we actually start using these for biomarkers for relative risk of disease, right? Cardiovascular disease, 25 to 50% of risk for cardiovascular disease is not predicted by just simply LDL cholesterol and other risk factors. So the last couple slides are just sort of the structures that when TM talks about these, you can flip these up and actually take a look, because he's going to talk about a lot of these different structures, and Tanu is going to talk about how she quantifies them, but at least gives you an idea what these are, and so you can actually have an image in your brain. So I won't belabor you. The one that I'd just like to point out is the cardiolipids. It's highly enriched in mitochondrial membranes, and so the cardiolipids and alterations in that seem to be, may give you some indication of what's happening in your in the mitochondria, and there's some evidence that the different fatty acids that are in the cardiolipids may affect the coupling and uncoupling state and efficiency of the mitochondria in different dietary and perhaps pathological conditions. So there's lots of them out there. The test will be cover all these when at the end, and I'll stop there. Is there any questions? I know it's just an overview. I just wanted to make sure that everybody sort of had an idea, or at least had something in front of you when you actually start hearing about these lipids. Because again, I've been using more and more of what TM's been generating, and it's really cool data. I think there's going to be a lot. I think this is going to be the really important thing to do in biomarker discovery, or at least one of the important platforms to use for that kind of thing. All right. Then, without further ado, I will introduce my friend and colleague, TM Rajendran.